The broadcast is now starting. All attendees are in listen-only mode.
Good morning or afternoon, and welcome everyone. I'm Christy Sullivan. I'm the Secretary of the American Society for Cellular and Computational Toxicology. And welcome to today's webinar presented by ASCCT and the European Society for Toxicology in Vitro. If you're not familiar with our organizations, both our societies host annual and biannual meetings and pursue other activities to promote the computational and in vitro toxicology fields, and have a particular focus on supporting early career scientists and offering training and education. I wanted to briefly give you a preview of some upcoming activities. First of all, we have 2 webinars coming in the next couple of months. You can register on our website, ascctalks.org/webinars, for the September webinar. The August webinar will be up shortly. So hopefully you will join us for those. And we also have an annual meeting in September, and there's still some time to get in a poster abstract if you're interested. It's going to be in Gaithersburg. The focus is on computational toxicology, but in vitro is well represented. So please feel free to join us. Really some brief housekeeping first. The webinar is recorded. ASCCT and SDIV members will have access to the recording. And everyone will receive a copy of the slides, a PDF of the slides. Your lines are muted to cut down on the background noise. If you have a question during the presentation, feel free to enter a question into the question box, and then once the presentation is finished, you'll be able, I will ask your questions to the presenter. You could also raise your hand if you want me to, if you want to ask your question directly. So in the control panel on the GoToWebinar screen there, you will see both a way to raise your hand and also a question module. So please feel free to enter your questions there. Our speaker today is Paul Watkins. He is the Howard Q. Ferguson Distinguished Professor and Director of the Institute for Drug Safety Sciences at the University of North Carolina. Dr. Watkins founded and chairs the Scientific Advisory Board for the DILIsym Initiative, which is a public-private partnership to predict liver safety of new drug candidates. And his presentation today is using quantitative systems toxicology, improving the safety of drugs while reducing animal testing. And I will give him control of the slides in just a moment. Okay, Dr. Watkins, you should be all set to go.
Okay, so Christy, people are seeing my slides right now?
Yes, that's right.
All right, well, I'm very pleased to be asked to talk about this subject, which has become close to my heart. By way of disclosure, I do chair the Scientific Advisory Committee for this DILIsym initiative, and I have a financial interest for another 10 months in the success of the spin-off company, DILIsym Services. So, by way of just introduction, this is one company's experience with why drugs fail due to toxicity or safety, AstraZeneca, but I think it's more or less representative. And the red bars are the number of percent of compounds that die in preclinical evaluation, and then the tan bars are during clinical evaluation. And what you can see is cardiovascular remains the major toxicity that limits developments of drugs. And of course, that's a big category. You've got prolonged QT, arrhythmias, you've got cardiomyopathies, you have atherosclerosis, and of course, there's a path in terms of healthy volunteer, prolonged QT studies, and now post-marketing requirements for some drugs to have very large long-term cardiovascular outcome studies. I'm not going to talk about that today, but there's quite a few efforts to apply QST to really help improve the detection and management of this problem. FDA has one project, but also private companies. The second most important, I think, still toxicity is liver, and that's because liver failure and death or transplant is really the outcome. And you can see that's close second to cardiovascular, and I'll be talking about that today. I'll also mention at the end kidney, which we're really talking about chronic loss of kidney function or acute kidney failure, need for dialysis and transplant. So my outline is I'm going to focus on liver safety with a brief overview in a little more depth of the issue, and then the current process that companies go by to assess safety of new drug candidates, talk about then the progress that's been made with this one QST modeling effort, which is DILIsym, and then some conclusions. So now you could look at liver toxicity and say that from a regulatory standpoint, there's not much of a problem because no drugs have been approved in the last decade. It's actually more like 15 years that have been withdrawn from the market due to unexpected liver safety concerns. And in part, I think this does reflect that people are smarter now, drug developers, and there are some improvements in the traditional methods to screen and assess liver safety. And regulators have found that there's a way in clinical trials to pick up this liability. But unfortunately, that method to pick up the liability inevitably means clinical trials get much bigger and longer at the first sign of a liver safety concern, and I'll be talking more about that. And just for an example, then and now, Resline or troglitazone was approved for the treatment of type 2 diabetes with less than 1,000 patients actually treated for 6 months. And now, of course, it would be thousands just to get consider approval, and then the post-marketing cardiovascular studies, et cetera, in class. But another example is rivaroxaban, oral anticoagulant that had an in-class concern for hepatotoxicity of the initial zymeligatrin being withdrawn from worldwide markets. There were actually 60,000 patients exposed to the drug before it received approval. And I just picked out this recent news just within a little more than a month. Motif Bio shares plummet after additional Icloprim clinical trial blow. Icloprim is an antibiotic that I think it could be a addition to meet an unmet medical need. But with this news release on Thursday, Motif explained it received the official minutes of its meeting with the FDA, and an additional clinical trial will be needed before granting marketing approval to address continued concerns of the regulator about potential for liver toxicity. And in general, when this kind of comment comes back from the FDA, you're not talking about small clinical trials. And there's a thing called the rule of 3. So if you want 95% confidence to exclude a serious liver event in 1 in 1,000 people, you need a trial of 3,000 subjects. And if you want to exclude more than that, obviously it goes up. And actually with an antibiotic, a company was told at time of NDA, or request for NDA approval, initially they would need to do a 12,000 patient study before liver safety could be adequately assessed. And the economics of that delay, it's obviously very expensive to run these trials, and but the major issue is actually the delay and years off patent. So if a drug attains at least $1 billion of sales at the end of patent life, which is a reasonable target for any new drug candidate in development, every day that you don't get FDA approval can equate to more than $2 million. So this translates not just into delay in delivering new drugs for people, but obviously driving up their costs. So drug-induced liver injury remains a major problem in drug development. It's driving up drug costs and delaying release of new drugs. And I'm just going to mention, this is also a problem for herbal and dietary supplements, where these things get on the market without sort of rigorous safety testing. And in their case, the benefits are generally not established. And this just makes the point in the U.S. Drug-Induced Liver Injury Network in this recent review of it, that actually herbal and dietary supplements was the second major class of drugs that were causing drug-induced liver injury in these people that are found and put in the registry. And now about 1 in 5 new cases of clinically important liver injury that enter the network are actually due to dietary and herbal supplements. And again, these are things generally that don't have any proven health benefit, and yet there are these risks. And I've shown this slide before, some of you may have seen it. It says, in only 6 weeks of drinking fit tea, Robert lost $500, which sort of brings home the point of unproven benefit. So the current process to assess liver safety of new drug candidates. Well, they're preclinical, and people are sort of going to the term non-clinical since you're going to man earlier and earlier, and these non-clinical studies continue into the clinical development. And there are and have emerged a variety of molecular screening tools that I think have been helpful in reducing liver safety liabilities. And this includes just the properties of the molecule. I don't know if people know, but rule of 2 and rule of 3 and this BDDS classification I'm going to mention a little bit. There's also more commonly screening, high throughput screening for off-target effects of drugs. And I think, and all this is, and looking for structural alerts, et cetera, and all this is helpful. But still, the requirement is to do animal toxicity testing, both rodent and non-rodent, usually rat and dog, that are quite extensive and both short-term, long-term, varying dose studies. And obviously, it would be very desirable not to have to sacrifice so many animals. And of course, in Europe, there's legislation that's really pushing this direction. And the problem is that we know that in spite of all these current tools, drugs will still go into clinical development and have surprises in terms of liver safety. But I think an unrecognized problem too is that often these give false positive results, and that actually safe and useful drugs are abandoned early in development that would in fact be safe had they gotten into people. And so a good example of that is acetaminophen. We all know that this is a problem with overdose, but I would venture that even if it treated serious disease, Alzheimer's or inflammatory bowel disease, it would never make it into man because you'd see dose-dependent necrosis in animal models. And in fact, when you go into people, you see evidence of liver safety, even at therapeutic dosing. And then ibuprofen, obviously widely used, Motrin, called vitamin I in the clinic because it seems like everybody's taking it. One of the safest drugs ever developed. But if your dog has even eats a few tablets, they are in serious risk of kidney and liver failure. So that's an example of a drug. Had it been developed at a time when the dog was a model, would not go into a man, even for serious unmet medical needs. So then the other thing is then, okay, you do the best you can, but then you go into clinical trials, and this is really where the FDA and this has close scrutiny in clinical trials. And what has followed is the serum alanine aminotransferase, and it's universally used really to detect and monitor liver injury. And this is a protein that's inside liver cells that leaks into circulation during hepatocyte injury or death. And it is a good liver marker as long as there's no muscle injury. The problem is there are false positives with this test. So drugs can cause elevations in ALT, yet rarely be associated with clinically important liver injury. And that would include the statins. It turns out cholestyramine, heparins, an earlier drug tacrine, and there are others where the signal that you get in a clinical trial in terms of these liver chemistries way exaggerates the actual risk, liver safety risk to the patient taking the drug. And this is because ALT, and furthermore, ALT elevations generally resolve with continued treatment, even for drugs that can cause liver failure. So even if you take a drug like isoniazid that clearly can cause acute liver failure, the vast majority of people taking the drug, you'll see the ALT elevations, but they'll go away. So ALT elevations occur with drugs that in fact are relatively safe for the liver, as well as transient elevations occurring with drugs that can be life-threatening. So how do you tell the difference in serious liver injury potential when you see ALT elevation? And the approach, which is codified in the 2009 FDA guidance on assessing liver safety in clinical trials, which still remains the most current guidance, is to continue to treat through the elevations as long as the patients don't get sick or symptomatic, to see if global liver dysfunction occurs, that is a rise in serum bilirubin, and what's called a Hy's Law case. So when you see these elevations, don't stop as long as the patient seems okay. Continue, but wait to see if there's some evidence of liver dysfunction manifested by a rise in serum bilirubin. The problem is, again, with the rule of three, you need large clinical trials to define the risk, and hence the requirement for large clinical trials once any liver safety concern arises. And this is actually the way liver safety is now the primary way it's evaluated in clinical trials. And I'm just going to walk you through it because I'm coming back to it with the QST modeling. What's shown here is the peak serum ALT that individuals had during a clinical trial versus the peak total serum bilirubin that they experienced during the clinical trial. So every one of these points is an individual patient. And in this case, the patients receiving the non-hepatotoxic comparator are the green circles, and those receiving active treatment are the red triangles. And the graph is divided into 4 quadrants by elevation in serum ALT greater than 3 times the upper limit of normal, the traditional cutoff for clinical significance for ALT elevation, and the elevation in serum bilirubin 2 times the upper limit of normal. And in this particular database, which was large and where active treatment and non-toxic comparator were 1-to-1, you can see that the vast majority of patients receiving the comparator had peak serum ALTs and bilirubins within the normal range, or at least less than 2 times or 3 times the upper limit of normal. Whereas those on active treatment had disproportionately developed elevations in serum ALT in this quadrant, also called Temple's corollary quadrant or range. And some of them then experienced bilirubin elevations while they were having ALT elevations. So you can now, with software in clinical trials, you can actually follow in real time whether patients are, what's happening to patients. And as I mentioned, the majority of patients that develop ALT elevations have them return to their baseline in this process called adaptation. And the only way to tell adaptation, that means everyone's going to be okay, or the vast majority, from elevations that can portend serious liver injury, is to see if individuals go here and then go up here. Whoops, sorry. In other words, enter what's called the Hy's Law range or become Hy's Law cases. And this is a traditional way that liver safety is evaluated in clinical trials, at least as a starting point. And this has now been incorporated into the QST model that I'll be showing you. So getting on to the QST model, DILIsym. Just background, why an academic hepatologist got involved with quantitative systems toxicology. I became director of a new institute, the Institute for Drug Safety Sciences, where we're really interested in what causes liver toxicity in patients. And we developed in vitro lab-based approaches to study mechanisms. Also have had a major program now headed up by Mary Mosdale looking at mouse genetics and what are called mouse genetic reference populations. And then out of that arose this public-private partnership called the DILIsym Initiative, which has been up until the end of last year supported by the FDA, actually funding an ORIS fellow who was an embedded member of the team. And the reason I got interested in this is it turns out, from experience I had with another project, that if you're forced to build these quantitative systems models, you take the data that's in the literature and you put them into the models, and it becomes very clear where the data gaps are. And it really becomes a very effective tool to guide, prioritize the research agenda. And so this was now started, almost 10 years ago, and what's come out of that was a spin-off company, actually, at the request of the partners. These are some of the current partners. Some companies don't like being advertised in this way, now that there's a sort of commercial aspect to it. And what we've benefited right from the beginning is an outstanding group, I think the world's experts in various aspects of understanding a drug-induced liver injury, including the mechanisms behind it. And I think, sure, some of you on the line know these people. And what's been done is to build what are called mechanistic modules using differential equations. And these are differential equations that recapitulate processes in the liver whereby drugs can hurt the liver, like glutathione depletion, for instance. And then as you build this model, it prioritizes the research agenda and identifies gaps, and therefore you prioritize experiments to fill in the knowledge gaps. And then these modules are integrated with the outcome of hepatocyte death and release and clearance of traditional and now more novel biomarkers, GLDH, miR-122, et cetera. And then the parameters in the model are varied to create simulated patient populations. And actually patient populations that differ, so healthy volunteers, diabetes, NASH patients, based where possible on the existing data that's in the literature or otherwise provided, say, by the partners, but also varying the parameters to recreate what's actually observed in patient populations. And then the model has been refined over the last years through incorporation of data from successive, what are called exemplar drugs, some from the literature, some unpublished data, quite a bit from our partners in terms of their own experience with drugs. And so this is a 30,000-foot view of the different modules that are currently in DILIsym. And of course, you've got drug metabolism disposition, you've got reactive metabolite, reactive oxygen species, mitochondrial dysfunction, alterations in intracellular bile acids. Very recently, cholangiocyte injury by one mechanism has been added. And all of this really is put in the model and integrated to predict the serum biomarkers over time as a reflective liver injury. And one of the things that's come out, which has been a surprise, is just 3 processes that you can measure in human expressed proteins or in human cells appears to account for the vast majority of dose-dependent liver toxicity observed in both rodents, dogs, and man, of course, the major focus being on man. So reactive oxygen species or oxidative stress, mitochondrial dysfunction, and alterations in bile acid homeostasis. Those 3 mechanisms, if you put the data in along with exposure into the model, is predicting about 80% of liver safety liability. And the outcome is then liver site death, but regeneration is built in the model, and the output is the various serum liver chemistries, including ALT, as a function of time. So the way this is actually done is you have to estimate the exposure of the liver to drug and major metabolites, not just outside, but actually inside the cells. And there's a variety of ways to do that. PBPK, if a mass balance study's been done in animal species, you can actually look at the radioactivity distribution between blood and liver. But then the ability to inhibit various bile acid transporters can be measured. In general, that's been done by a company called Solvo that I have no financial interest in. Then mitochondrial respiration and reactive oxygen species is generated in liver cells, variety, initially HepG2 cells, now moving to human hepatocytes, spheroids, HepG2, using the Seahorse instrument and high content imaging. And then the parameters are varied to create these simulated populations. And when all this information is added into the model, what comes out is a simulated frequency and severity of liver injury. So the example I'm going to give you, which is relevant to species differences, has to do with drugs causing toxicity by altering bile acid homeostasis, leading to hepatocyte death, regeneration, and serum biomarkers. So this is an Amgen compound called 009. There's no evidence of liver injury in multiple species. Interestingly, rats, mice, hamsters, rabbits, and non-human primates. Now, I believe the hamsters, rabbits, and non-human primates were actually done after the fact, just out of curiosity, whether they would actually show the toxicity. But during phase 1 dose escalations in healthy volunteers, 5 out of the 8 patients showed significant, unfortunately reversible transaminase elevations that was dose dependent, and the further development of this drug was halted. And then there was a series of investigations trying to learn from this experience, and inhibition of 2 transporters, and maybe 3, were found as the only potential liability. That's BSEP and MRP3 and MRP4. There were no reactive metabolites, no covalent binding, and no mitochondrial toxicities. So what this means is that shown here is basically a liver cell. The beginning of the biliary tree is the bile canaliculus here. Bile acids are taken up into the liver predominantly by a transporter called NTCP, and then they're pumped out of the liver cell into the bile by this transporter BSEP, but can also efflux back into circulation through MRP3 and MRP4. And what they were saying with AMG 009 was that it inhibited both the transport of bile acids into bile and the efflux mechanism, which would lead to increase in bile acids, and bile acids can cause toxicity. Now, it's not totally straightforward because most drugs that inhibit the hepatocyte bile acid efflux transporters also inhibit the uptake transporter to some degree. So this is really an ideal situation for modeling to figure out what the net result of all this is. So what was done is the transporter inhibition studies and data, the inhibition constants were put in the model. They had enough information to estimate liver exposure, and then the healthy volunteer SIMPOPS was used, since that was what was the population in the phase 1 trial. The simulated frequency and severity of injury was estimated. So what's shown here is the time on treatment versus the time-dependent elevation in serum ALT, and the black symbols are the actual values in the clinical trial. And you can see you were getting a significant, this is full baseline, significant elevations in serum ALT that peaked somewhere around 1 week on treatment. And then in red is shown what the simulation showed in the simulated healthy volunteer population. And you can see it clearly was predicting ALT elevations, and actually the mean elevation was also about 1 week or maybe a little longer. And furthermore, the actual dose-dependent elevations were fairly well predicted by the DILIsym model here. The predictions are in red and the actual values are in black. So DILIsym predicted the dose-dependent delayed presentation of AMG 009 hepatotoxicity and recovery after discontinuation. And the incidence rates and the timing, actually, of the elevations were very similar to what was actually shown in people. Now, when they did the same thing in the rat DILIsym model, there really was no toxicity, only tiny little blips, and basically reproduced what was seen in the animal model. And this reflects, of course, interspecies differences to some extent in the ability of the drug to inhibit the various transporters. But also, we now know that the rat and other preclinical species profile of bile acids is inherently less toxic than human bile acids. And now I think we appreciate that a major reason for the occasional discrepancy between rats that is showing no toxicity and suddenly seeing it in humans is likely, or at least a likely cause, is bile acid alterations in homeostasis. So the backup compound they moved right into clinic. Again, there was no evidence of liver injury in preclinical species. And with this drug, they saw no liver toxicity in human studies when they started them. And this was odd because in retrospect, again, it was after the fact, they saw that this backup compound, AMG 853, was actually a more potent BSEP inhibitor than AMG 009. The IC50 was 4.3 micromolar versus 11.5 micromolar. So had they done the BSEP inhibition studies first, they would have been more concerned about the backup compound that in fact was safe in humans than with the AMG 009 compound. And actually, DILIsym modeling for AMG 853, the follow-up compound, was safe in the simulated patient populations. There were no ALT elevations that were shown. Now, the exposure is lower, but even when they raised the doses to simulate 50-fold clinical doses with AMG 853, it remained safe. So it was an inherently safer molecule than AMG 009. And why was that the case? Well, it turns out, and this is a major insight that's come out of this public-private partnership, the DILIsym initiative, is that all inhibition of bile acid transporters is not the same, and the type of inhibition is key in figuring out the implications of inhibition. So, for instance, with AMG 009, as I said, it inhibited both MRP3 and MRP4, but it inhibited BSEP as a non-competitive inhibitor, essentially knocking it out, so that as bile acids increased, they caused hepatotoxicity. Whereas with AMG 853, it was a competitive inhibitor. So as the bile acids increased in concentration, they were actually able to outcompete the inhibitor, preventing elevation to toxic levels in bile acids. And so again, this is an insight into the complexity of interpreting BSEP inhibition. Now, before I make another comment about BSEP inhibition, let me just say, so QST modeling was able to predict species differences in hepatotoxic potential of AMG 009 and the safety of AMG 853 in man based on in vitro assessments of bile acid transporter inhibition. And that's despite the fact that the BSEP inhibitory concentration 50 and Ki actually were lower for 853 than for 009. And I won't talk about this at all, but this actually came out just last year, a collaboration between the DILIsym group and AstraZeneca, Jay Medahall and Simone Stahl. But in this, it's interesting. They talked about that a dose was toxic in rats, but the single doses were not toxic in a simulated population, and that fit their clinical data. There was toxicity in the rat, but when they went to man, they didn't see toxicity. And species differences were largely attributed to difference in liver exposure, but increased sensitivity to inhibition of mitochondrial respiration in the rat was also contributed. So what DILIsym is showing is explaining species differences, one in bile acid homeostasis, but also in this case, differences in the ability of the drug to actually inhibit mitochondrial respiration. So another important insight. Now, I thought I'd just mention this. My good friend Les Bennett, and I mean that, I've known him for a long time, has been basically poo-pooing BSEP inhibition as a useful thing to measure in terms of predictors of DILI. And he's particularly touting classification of molecules based on physical chemical properties, and pointed out that what's called class 2 BDDCS drugs, have very high liver safety liability, and we could solve the problem basically just by avoiding drugs that have those characteristics. And I thought I'd just show a comment from Will Proctor, who's a leader, no question, in preclinical liver safety evaluation. And he wrote, we're targeting harder proteins such as GPCRs, ion channels, and previously undruggable targets by targeting them for degradation or affecting protein-protein interactions. These new molecular entities are all typically BDDCS class II molecules with high permeability, relatively low solubility, and high metabolism. They also have a chance of hitting BSEP and other bile acid transporters, affect mitochondrial respiration, and by metabolism alone, higher chance of forming reactive metabolites. Sometimes potency tracks with certain physical chemical properties that build in higher risk. BSEP screening has enabled us to progress and characterize the risk without killing molecules thoughtlessly. We could do the same BDDCS class II, do the same for BDDCS class II molecules, assume there is higher DILI risk. However, it doesn't give us something to inform on mechanisms, a counter screens used for backup molecules, or to help monitoring strategies. And I think the point is that, obviously the dose makes the poison. You really wouldn't want to exclude a drug that was a rare molecule that was effective in hitting your target simply based on these properties if you could get an effect at a dose that was non-toxic. So I'm going to tell you about one modeling exercise, and there have been others like this with a compound. The identity can't be revealed yet. It was initially developed for one indication, but during the early clinical trials, they actually had a Hy's Law case, this elevation of bilirubin, and they terminated the program. But they kept the molecule in their high throughput screening assays and found it was a very potent molecule in terms of interacting with another target that might be very valuable in another disease. So the question was, could lower dosing of this new, in this new disease indication be safe? And so again, all the data was put into DILIsym, and the results looked at. And I'm going to show you the results can be shown in DILIsym as it can actually predict what the E-dish plot is going to look like. And so when they looked at the prior clinical trials where Hy's Law cases were shown, there were 3 doses that were used. The 0.3 times dose was generally safe, although there was one simulated patient that had an ALT elevation greater than 3 times the upper limit of normal, shown by my pointer here. At 0.5 times the dosing, there were Hy's Law cases as well as 1 times. So it predicted the liability of the molecule at the doses that were initially used for the initial indication. However, when they modeled a much lower dose, which was believed to potentially be effective in the second indication, there were no elevations in serum ALT. And this data was presented to a division at a face-to-face FDA meeting. And it's my understanding that the development of this drug for this new indication is going forward. So at lower dosing regimens proposed, compound Y may have an acceptable liver safety profile for the new disease indication. So this is a list of the known DILIsym applications that have been submitted to or intended for regulatory agencies. I believe the list is a year old and has increased since then. The FDA itself has acquired a license for the software and actually renewed it this year. And I think it's fair to say that the FDA is interested in this modeling as part of the aggregate data once a liver, to assess a molecule safety once some liver safety concerns appear. So one of the neat things about the modeling is you can actually use it to develop mechanistic insights. So again, we have all our parameters here. And you can actually analyze mechanisms. And you do this by just in the model turning off one mechanism and turning off one at a time sequentially. And just seeing what it does to the frequency of modeling if in the simulated population. So if there's no effect on the predicted frequency of ALT elevations, when you turn off one of the mechanisms, then that's not contributing. On the other hand, if one mechanism, say mitochondrial respiration, is accounting for the majority or all the toxicity, that turns out to give you insight into patient risk factors. And there's one example of a drug where the DILIsym predicted the toxicity, and it was mainly due to interference with mitochondrial respiration. And it was noted that patients taking metformin, a drug that also interferes with mitochondrial respiration, there was an increased incidence of ALT elevations. So I think part of the future is using this kind of information to actually inform risk management. So understanding mechanisms can identify patient risk factors. So I'm getting to the end here of talking about DILIsym. Of course, this is the standard preclinical to clinical phase of development. And most of the applications of DILIsym have actually been early in clinical trials. I gave you an example of dose alteration or optimization, infer the magnitude of injury based on measured biomarkers, extrapolate from healthy volunteers to individual patient groups in that they're simulated populations for different patient groups, and then a guide incorporation of emerging biomarkers into clinical trials. I didn't talk about that, but that is an area of particular interest to me. And also now when signals are being picked up later in clinical trials to identify risk factors that I think can lead to personalized medicine approaches to risk management. And what we're now seeing, which is more relevant maybe to the talk here, is really moving this into preclinical assessment to rank candidates for DILI potential and to understand discrepancies that may occur between animal studies and in vitro findings in humans. So I was told to add a slide, how can you get DILIsym? Well, if you're in academics, there are low-cost licenses available, and several academic centers have acquired licenses, something I'm very enthusiastic about. I think obviously the more people are using this, the better. For industry, you really have two options. You can join the DILIsym initiative. It's $100 grand a year, but then you have training and you have input into the actual direction of the modeling. Or there are now new licensing operations, and I think you can go to the DILIsym website or contact Brett Howell, who's the president of the spin-off company DILIsym Services, at bretthowell.dilisym.com. So I said I'd mention kidney. Well, it turns out at the request of our partners, the team has now embarked on applying QST to kidney injury, and we're fortunate enough to receive a $1.7 million phase 1, phase 2 grant. And this is starting now a new consortium, a RENAsym consortium. And Merck has been the first to join. They're very interested in this area. They've agreed to share data on compounds they have that have shown species differences in kidney toxicity. And also data they have with newer urine biomarkers. And Frank Sester is going to be directly involved with this. So very excited about that. Very low cost. I think it varies, but very low cost to join because the effort is largely funded. And we're looking for new partners for this. We again have a first-rate team of advisors. People in this field will recognize all these people as absolutely the top international figures, except for me in kidney toxicity, but I'm tagging along. And the plan for the very first version it has been mapped out, and modeling is now underway, looking at human and rat. I think this will be an exciting new development that we can all sit and watch how it comes out. So conclusions. I think QST modeling is already having a significant impact on decision-making and drug development. And certainly we're seeing wider and wider applications of DILIsym modeling and incorporation into regulatory communications. And I think its applications should and will lead to reductions in improved safety, of new drugs, smaller and smaller clinical trials, and I think it should lead to a reduction in animal testing. And it's too bad that Scott Gottlieb has stepped aside because he was a great advocate, and this was shortly after he became commissioner, talking about the CEDAR using modeling and simulation to predict clinical outcomes, inform clinical trial design, support evidence effectiveness, optimize dosing. predict product safety, and evaluate potential adverse mechanisms. And I think Ned Sharpless would also be enthusiastic. He's from UNC. I know him personally. And we'll be putting out additional updated guidances on how aspects of these in silico tools can be advanced and incorporated different aspects. And that is, I think, still ongoing. A series of guidances are in the pipeline. So in closing, I want to acknowledge the DILIsym team. They're really a fantastic group of people, really led by Scott Seiler and Brett Howell. They have open positions for those out there that are interested in entering this exciting field. It's very hard to find the right people, but if you're interested, give them a call. So with that, I'll quit, and we have more than 10 minutes for questions.
Great. Well, thank you very much again, Dr. Watkins, for your presentation. And I have a couple of questions coming in. I see. I'll start reading them out. And I saw someone's hand go up and then come back down. So if you do want to raise your hand, keep it up and I'll be able to call on you. So the first question is, how is ROS, reactive oxygen species generation, modeled in the DILIsym module? So I guess they're just asking how that's modeled. Yeah.
That's above my pay grade, I'm afraid. In other words, the data is collected either in-house at companies, and of course the assays have been picked by our partners because they are either in-house or commercially available. And the usual process is to look at ROS generation through and glutathione depletion through high content imaging. But how that data, the concentration dependent effects, and they measure both, the nominal media concentration and actually mass spec the concentration inside the cells. How that's translated into the model, I'm not an expert on. And unfortunately, only I was asked to give this presentation.
That's okay. We have a great question. How can this model be applied for biologics?
Yeah, that's a really good question. It hasn't been voted on as a high priority by the partners, although I always keep hoping it will be. DILIsym Services was acquired by Simulations Plus, who have, PBK software for, and in the process of integrating it in with DILIsym, their GastroPlus, and have, quite a bit of experience with biologics. And there's no question that biologics, can cause liver injury. And right now, a major area actually is in bringing in the adaptive immune system. And we're fortunate enough, Liesl Schoda right here, who came from Enelos, has a lot of experience in this area and has been leading the team, bringing in adaptive immunity. And what they're doing is they're moving towards modeling checkpoint inhibitors. Of course, monoclonal antibody would be one example of a biologic there, and figuring out how reducing immune tolerance, will lead, and that's with Jack Utrick and his PD-1 knockout CTLA-4 antibody model. So that's an example of an area where biologics are being explored right now. But I think if we had a company or companies that came into the initiative and really pushed the biologics, we would be very excited to do it, to get really roll up our sleeves and get involved with that. Whether it's on target or off target effects, we'd be very interested in that.
Great. Have there been cases where the model did not predict Hy's law, I guess, observed in the clinic?
Not in clinical trials. The model didn't predict the liver safety liability, for instance, of Ketek, telithromycin. And that's a drug that was essentially withdrawn from the market because of post-marketing cases of severe liver injury, Hy's Law cases. Now, they were extremely rare, had to be, but that's an example where the model didn't predict the post-marketing liability. And I think that's an important point, is that, you know, the model is still in evolution. In other words, the initiative is continuing, the model's being constantly improved and changed. And that's why, you know, it has to be part of the weight of evidence in decision-making rather than relying on it, you know, totally. But to my knowledge, there's been no drug that in clinical trials caused a Hy's Law case that was predicted to be safe, for instance, by DILIsym. Ketext's the only example that comes to mind. There may be others, you know, because I'm full-time university, I have to keep arm's length from the DILIsym services business. That's the only one I'm aware of.
Okay. Yeah, some very sort of complimentary feedback about great work, and also some questions like that about the limitations, which I think you just kind of responded to, so that's great. So question about this discrepancy in mitochondrial impairment in DILI, which I think you sort of addressed in your talk, stating that it's an important mechanism, but other folks not thinking that it is. Do you know what's behind that?
Yeah, I can address that. You know, sort of the skeptic of the role of mitochondria in DILI is Jack Utrick, you know, who talks about it quite a lot and is on our scientific advisory board, as you saw. So we're well aware that he feels that there isn't solid data, and when in his model he tries to intentionally reduce mitochondrial function, he doesn't see an effect in his models. But I think the overwhelming evidence is that, and I think DILIsym modeling is perhaps the strongest example of that. And the fact, you know, the interaction, I say, with metformin, for instance, and ALT elevations in one drug. I think the evidence, from my perspective, is overwhelming that interference with mitochondrial function is one of the mechanisms that underlie drug-induced liver injury. Now, the liver is a clever organ, and this adaptation, we know that if you inhibit mitochondrial oxidative phosphorylation, the liver responds by making new mitochondria called mitogenesis. So it will, and that's actually in DILIsym now, is the effect of mitogenesis if you reach certain thresholds for ATP loss due to interference with mitochondrial function. And you can show actually the resolution with continued treatment with drugs that have that as a major mechanism. But it's still a mechanism. And I think, you know, you can have trouble before the adaptation occurs, the mitogenesis, and also, you know, people probably regenerate mitochondria at different rates. So I think that the overwhelming weight of evidence is that interference with mitochondrial function is a mechanism that's relevant for drug-induced liver injury.
Mm-hmm. Okay. There's a question about using this model for fish or other ecospecies. Has there been any discussion of that? Sounds like sort of a fair departure from.
No, there hasn't. You know, rat and man is well developed.
Yeah.
Dog, unfortunately, there isn't a lot of data, but you know, the attempts have been made to modify the model based on what data exists. And monkey, you know, would be great. No one has requested fish yet. And the issue would be, is there a demand for it? Mm-hmm. And is there the data available to actually do the modeling?
I think probably they're thinking in the direction of, you know, maybe drugs in the water system and maybe that.
Oh, yeah, right.
Yeah.
Sure.
Great. Okay. Okay. The fact that the variability that's seen in the data is incorporated into the predictions is great. What about potential variability that could appear outside of the data set, which remains unknown? So unknown or undetected variability. How do you account for that?
Right. Well, the goal, is to make these simulated populations that really accurately reflect the extremes of susceptibility that would be in far larger populations, to the point that the equivalent of a 12,000 patient clinical trial or something. And the populations, simulated populations have been built based on available data in terms of variation. So for instance, NASH patients, there's a liver biopsy study that shows they have reduced enzyme activity in the electron transport chain, and that's put in the model. But then also the population's parameters are tweaked to give you the results that, or have been tweaked over time to be consistent with the results that have been observed in large clinical trials. But again, there's no guarantee that every type of individual is in there. And what's very exciting now is there are several large clinical trials that are ongoing, in this case, next-in-class drugs that had problems with first-in-class, that are testing DILIsym's prediction that they'll be entirely safe for the liver. And the results of those are going to be coming out within the next year. And obviously that would be great if it really predicted in a 3,000 patient study that no one would have a significant liver injury. But it's too early to be confident about that.
Mm-hmm. Okay.
I might add too, one of the interesting ideas is, because there are always exclusion criteria, shouldn't it be possible ultimately to not have to do a study in NASH patients or diabetes patients or heart failure patients because the modeling says that it would not be relevant, just as you don't need to do drug interaction studies because modeling and simulation based on microsomal P450 inhibition studies say that there shouldn't be a drug interaction. And that's a ways off, but I think that will come. And again, that will reduce the costs and improve the efficiency of drug development.
So one last question, and it's about interaction with cardiovascular, so drugs causing adverse reactions in multiple organs. And so is there a way to align DILIsym with the CEPA initiative, for example?
CEPA. I should know what that is.
Yeah, I don't know exactly what it stands for. That's
not the microphysiologic system.
No. It's modeling for cardiovascular.
Oh, cardiovascular, yeah.
Events, yeah.
I'm not sure. Obviously the big hype around the multiple physiologic systems and the hundreds of millions of dollars have been pumped to create the human on a chip is a very attractive idea. And actually, the DILIsym team is collaborating with Lance Taylor at the University of Pittsburgh, who has one of the leading liver modules. But I think ultimately, to make the most sense out of those systems that are put together, it'll be critical to have quantitative systems, toxicology, and pharmacology be part of it to interpret it.
Yeah.
And that's a very exciting future area, I think.
Great. Well, I think we landed perfectly on the dot here. So thanks again so much for your presentation. Thank you all for attending and listening and contributing to the discussion. And I will hope to hear from you all and hope you'll join us at our next webinar. And I'll say good afternoon or good evening. Thanks.
Great. Thanks, everybody.
Thanks. Bye-bye.